Hello, I am Rosalino Vázquez López, a research at the Universidad Anáhuac, Mexico. My team and I appreciate the invitation to participate in this second edition of COVID-19 International Congress. One of my line of research is the study of pathophysiology of SARS CoV-2 infection. It is a pleasure for us, my team and I, to share today our documentary investigation entitled Neurological Complications After SARS CoV-2 Infection. Thank you.
Hi, good afternoon. My name is Cristian Parra, and with my colleagues Almudena Chavez, Max Metgasca, and Jimena Sanchez, with the support of the researcher Rosalino Vázquez López, we have the pleasure of present the following topic: neurological complications after SARS CoV-2 infection. As all we know, the SARS CoV-2 is transmitted by saliva droplets from an infected person. Then the virus gets into the lower respiratory system and infects the alveoli cells. The alveoli is conformed by two types of cells: the type 1 pneumocytes, which are going for a gas exchange, and type 2 pneumocytes, which are going to produce surfactant so the alveoli don't collapse. In the type 2 pneumocytes, the S protein of the virus binds to the ACA2 angiotensin converting enzyme 2, which in this case is the receptor of the virus. ACA2 is a membrane protein that is present in different tissues like nervous system, endothelial tissue, kidney, lung, and heart, and so on. In the other hand, the virus has the spike protein, which is conformed by three subunits, each one with three domains, S1 and S2. S1 engages with the host receptor, and S2 mediates the membrane fusion and the viral entry. For the viral entry, the participation of the transmembrane protease serine2 is necessary. This protease mediates the cleavage of S1 to S2. Then the viral entry happens. The renin-angiotensin system is also important in the pathophysiology. As is known, this system is activated by local or systemic infection or sepsis. After the viral entry, ACA2 levels decrease. Thus, the levels of angiotensin 1 to 9 and 1 to 7 also decrease. As well, the levels of angiotensin 2 increase and binds to its receptor, which is the AT1R, having the following effects: vasoconstriction, inflammation, fibrosis, and so on. ACA2 can antagonize and produce the pathological process, but in this case, the enzyme is blocked by the S protein of the virus. So the effects of angiotensin 1 to 7 do not antagonize these pro-inflammatory effects of angiotensin 2 and its receptor. Talking about the immune response, the viral RNA is taken by endosomal TLR, the TLR3 and TLR7, and RIG1 that signalize to NF-kappa beta and IRF3. These transcription factors synthesize cytokines, contributing to the cytokine storm. The first line of defense against viral infection consists in NK cells, macrophages, dendritic cells, type 1 interferon, chemokines, and cytokines. Type 1 interferon is induced by PAMs that are mediated by PRR, like endosomal TLRs and RIG1. However, type 1 interferon was not detected in SARS-CoV and MERS-CoV infections. Moreover, SARS-CoV blocked the signalized pathway to IRF3 and 7. A recent whole blood transcriptome and cytokine profile indicated that type 1 interferon was low in several cases of COVID-19, suggesting the impairment of type 1 interferon antiviral response. The delayed type 1 interferon response and unrestrained viral replication promote pro-inflammatory process and lung pathology. These coronaviruses repress the induction of type 1 interferon by inhibiting the activation of IRF3. This allows a coronavirus to replicate unrestrainedly. While these type 2 pneumocytes produce pro-inflammatory cytokines and chemokines to mediate the recruitment of immune cells. Usually, NK cells suppress the viral replication by killing the infected cells. This by two ways, via granule or indirectly activating macrophages by releasing interferon gamma. Some factors that at the moment are unknown cause NK cells impairment in this infection. A dendritic cell presents the antigen peptide to a CD8 in a lymphoid organ. Now the CD8 kill infected cells by releasing granules, such as perforin and granzyme. Also, CD8s release interferon gamma. And CXCL10 for the CD8 migration. Alveolar macrophages and recruited macrophages get accumulated in the microvasculature of the lung. Due to the accumulation of the immune cells, the alveolar capillary membrane gets thicker, making the gas exchange hotter. Macrophages are activated by virus antigen stimulation, interferon gamma, or oxidized phospholipids. The interferon gamma and its complex induced the phosphorylation of STAT1 by JAK1 and 2, promoting the transcription of interferon-stimulated genes and pro-inflammatory cytokines and chemokines, contributing to the cytokine storm. Activated macrophages also release some toxic mediators, including inducing apoptosis. Yeah.
COVID-19 can present multiple symptoms such as fever, dry cough, fatigue, or short of breath. These symptoms are the most common ones. And if a patient, not all the patients may present all the symptoms, they can only present one or two. And of course, they can appear positive in the COVID test because they have the virus inside of them. And however, this disease can present other uncommon symptoms or unlikely symptoms, such as loss of smell, taste, gastrointestinal problems, and even neurological ones, such as confusion and disorientation. And whenever a patient presents these symptoms, these unlike symptoms, of course, those cases can be more serious than the other ones because these are rare ones, and all these kind of patients need to be attended immediately. However, COVID-19 may cause multiple complications such as heart failure, kidney failure, thromboembolism. That this one, of course, may be considered one of the most common complications by COVID-19. Liver complications, heart attacks, and of course, septic shocks. To avoid these kind of complications, all the patients that are positive to COVID-19 need to be, of course, attended immediately to avoid even to have any of the symptoms I mentioned before. To avoid to have this kind of complications, because once they have, for example, a kidney failure, of course they can be treated, but the recuperation time can be much longer, or it can be worse. Some of the patients that can have complications by COVID-19, they don't even make it. So if they start to have these simple symptoms, they have to be treated immediately to avoid these kind of situations.
Despite having many doubts about the neurological complications caused by SARS-CoV-2, some studies carried out have begun to show which have been the main ones. In the study carried out by Moreguchi et al., it was shown that patients could suffer from headaches, altered consciousness, seizures, and meningitis. In addition, it was possible to observe in a 34-year-old young man how there were affectations in the mesial temporal lobe, in the hippocampus, and in the lateral ventricle. The virus was identified in the cerebrospinal fluid. Another study, carried out by Mao et al., was carried out in a hospital with 214 patients, of which 36.4% presented neurological complications, which later it was observed that 24.8% was a central affectation, 10.7% was a peripheral affectation, and 10.7% was a musculoskeletal affectation, having dizziness, headache, hypogeusia, and hyposmia as the main symptoms. And the study by Helms et al. Showed that 84% of patients in intensive care for a severe infection had neurological complications, being encephalopathy, agitation, and confusion the main signs. And in the same way, the presence of Guillain-Barré syndrome was observed in 5 patients in Italy.
Neurological complications occur in severe SARS-CoV-2, and the probability of having neuronal degeneration or cerebral edema is high. Some of the most common complications are smell and taste disorders, encephalitis, acute necrotizing hemorrhagic encephalopathy, Guillain-Barré syndrome, and cerebrovascular complications. The anosmia, as we can see on the figure of the left, could be explained because of the virus produces a direct damage in the olfactory receptor neurons located in the olfactory epithelium. This is because the presence of 2 proteins found in the upper part of the respiratory tract, called AC2 and transmembrane protease serine-2. These proteins internalize the virus to the cytoplasm and are expressed mainly in suscepticular cells of the respiratory epithelium, becoming its main target. Likewise, the virus could produce a direct invasion of the frontal lobe, traveling through the axons of the olfactory nerve, causing a sensory neural alteration of smell. Similarly, a neurological study showed widespread tissue damage in neurons, glia cells, axons, and myelin sheds, associated with the presence of numerous SARS-CoV-2 variants, which became progressively more serious from the olfactory nerve to the rectus gyrus of the frontal lobe and brainstem. And on the right one, ageusia is due to the altered taste bud renewal after SARS-CoV-2 induced cytokine storm in distant cells, as well as a distant interferon production from infected keratinocytes, leading to AC2 expression in taste bud cells, which in turn may be infected by SARS-CoV-2. Another explanation could thus be that following infection of epithelial cells in the tongue, inflammatory cytokines could reach the taste buds, impairing their renewal. Indeed, toll-like receptors and interferon receptors are highly expressed in taste buds, and their activation may limit taste cell regeneration. Encephalitis caused by hypoxia due to infection of the virus generates an increase in anaerobic metabolism and metabolic acidosis, leading to headaches, alteration in consciousness, and cerebral edema. Acute necrotizing hemorrhagic encephalopathy is presumed to be the consequence of an intracranial cytokine storm that affects the blood-brain barrier, triggering an hyperinflammatory syndrome leading to fulminant and fatal hypercytokinemia with multi-organ failure. The neural damage of Guillain-Barré syndrome is caused by post-infectious autoimmune mechanism, whose response provokes the destruction of the myelin of the peripheral nerves, and in most cases is triggered by an infection. During SARS-CoV-2 infection, the virus binds this receptor and is endocytosed. Due to similarity in the peptide sequences or epitopes of SARS-CoV-2 and gangliosides, the antibodies formed against the virus through the T-cell B-cell interactions may bind the ganglioside located in the peripheral neurons. This may result in an autoimmune response that destroys the myelin and/or axons. The demyelination or axonal damage disrupts neuronal transmission, which causes GBS syndromes such as muscle weakness, paralysis, coordination problems, breathing difficulties, and autonomic dysfunctions. Finally, cerebrovascular complications are found in non-severe patients, and it's caused by the affinity of SARS-CoV-2 for AC2 receptors on endothelial cells, causing an increase in blood pressure. This increase, along with thrombocytopenia and coagulation disorders, contribute to the increased risk of both ischemic and hemorrhagic cerebrovascular accidents in patients with COVID-19. Similarly, cytokine storm syndrome may be another risk factor for cerebrovascular disease because of its relationship with damage to the blood-brain barrier. In conclusion, COVID-19 generates a state of significant neurological inflammation, attacking neuronal, glial, and endothelial cells through cytokine storm. Due to the above, neurological alterations were observed at the level of the peripheral and central nervous system. The production of cytokines that compromise the blood-brain barrier and the post-infectious outcome generate the damage. Likewise, we cannot rule out that patients may be more susceptible to triggering neurological disorders due to genetic or epigenetic antecedents. With the evidence studied, more future research is required to understand in greater detail the molecular mechanism involving the pathophysiology of neuroinflammation, especially before the appearance of new variants of the virus, such as the Delta, that seems to be more virulent. In this way, with greater knowledge, it will be possible to administer preventive treatments, such as immunizations, that consider neuropathy due to inflammation caused by SARS-CoV-2 infection. Merci beaucoup pour votre attention. Nous espérons que vous avez trouvé ce sujet intéressant. Salutations de Mexique.